Hey guys, last week we talked about the phases of meiosis and in particular prophase 1 and how unique it is. We knew that sexual reproduction allows for genetic variety, and that comes in two ways. Fertilization, which is the combining of genetic information from two different parent organisms, but also this unique process called crossing over. So last week we had this story time type of deal where we started with a cell that was a diploid cell. And this diploid cell has a maternal chromosome and a paternal chromosome. So it has one chromosome from mom and one chromosome from dad. And in humans, we have 23 chromosomes from mom, 23 chromosomes from dad. So in meiosis, we have interphase as well, just like we do in mitosis. And so those chromosomes are duplicated. And once those chromosomes are duplicated, because these chromosomes are homologous, meaning that they have the same information in the same order, they're able to connect and exchange information called through the process of crossing over that happens in prophase 1. So as we said last week, after they cross over in prophase 1, they line up in the middle for metaphase 1. The homologous pairs are split in anaphase 1, and we have two new cells with those sister chromatids in telophase 1. In prophase 2, the nuclear membrane breaks back down. The chromosomes line up in the middle and the spindle fibers attach. The sister chromatids are pulled apart, and cytokinesis occurs in telophase 2. And what happens is we end with 4 haploid daughter cells that are genetically different from the parent cell and from each other. So the overall process of meiosis, we started with a cell with 46 chromosomes, and we ended with cells with 23, and they're all going to be different from each other and from the parent cell. So what we want to do today is we're going to fill out this graphic organizer that highlights what the crossing over process is. It's important to know that crossing over is also called genetic recombination, which just means that we're changing the order of things within those chromosomes. So right here it says these are what kinds of chromosomes, and it's important to remember that crossing over can only occur between homologous chromosomes. Homo meaning same. So they have the same order of genes on those chromosomes. And that allows them to cross over. If we look at the center of these chromosomes, this is called the centromere, which we've talked a lot about. The centromere. And just like we saw in this original cell, we get one of those chromosomes from dad and one of those chromosomes from mom. So this is we'll just say that this one is the paternal chromosome, which means from dad. And this is the maternal chromosome, which means from mom. So this is the chromosome that came from the egg, which was then duplicated to go through crossing over. And this is the paternal chromosome from father, which was then duplicated in interphase for crossing over. So what we're going to do to symbolize this is we're going to use two different colors to show the two different kinds of chromosomes. So we have the paternal chromosome from dad, and we also have the maternal chromosome from mom. Okay. Now if we go to number 5, it says define homologous chromosomes. So we have to define what makes these two chromosomes homologous. And that just means that there are chromosomes that contain the same traits in the same order. And by traits here, we really mean genes, those sections of DNA that control traits. Meaning The end of this chromosome might control hair color, and same thing for mom's chromosome over here. Next little section might control eye color, same little section over here. Now dad might code for blue eyes, mom might code for brown eyes, but they're going to be in the same section of those chromosomes. Okay, so number 6 says, what are sister chromatids? And label these to the right. So sister chromatids, as we talked about in mitosis and meiosis, is this duplicated section of a chromosome. So it is each side of a chromosome connected by a centromere, and it is the copied info. So remember, if we go back to our original cell, we had the paternal chromosome, the maternal chromosome, and during interphase, that information is copied. So we just took mom's chromosome and we made an exact copy of it and attached it in the center at the centromere, and same thing for dad's chromosome. So if we look over here again, we still have dad's chromosome, or the paternal chromosome, and they are starting to link up. They haven't done anything yet. They're just lining up in interphase right now. Okay, so they are lining up and they are starting the process of crossing over. And crossing over is the exchange of genetic info between two non-sister chromatids of a homologous pair. So we have our homologous pair, and we have these two are sister chromatids, and these two are sister chromatids. So we have to take the non-sister chromatids, so one from this chromosome and one from this chromosome, and that's what we're going to cross over, is this two non-sister chromatids right here. So we're going to show that swapping of information. But over here we're going to color the paternal chromosome. But because of crossing over, we now have a section in our chromosome from the maternal chromosome. So that one little section there was swapped over in this process of crossing over. And same thing for mom's chromosome. We're actually going to have a small sliver of dad's genetic information on the maternal chromosome. So once again, that happened right here through the process of crossing over. Those two non-sister chromatids connected, swapped information, and then they're going to separate later on in anaphase 1. Now number 9 says, explain how genetic variation results from crossing over. And we know that this happens because we have this combination, or this exchange of genetic information. So exchange of genetic info creates 4 completely different chromosomes that will be divided among 4 daughter cells. So if you go back to the Amoeba Sisters video for meiosis that we watched a few days ago, she actually mentioned that you have the parent somatic cell. Now this doesn't mean parent as in reproduction parent, it just means a parent cell with 46 chromosomes, and it's going to create those 4 genetically different gametes. So this is a somatic cell, diploid cell, and we're making 4 genetically different gametes. So if this is all of one person's egg or sperm, each one of these has a different combination of genetic information because of that crossing over. So we have those 46 chromosomes, which just means we got 1 from mom, 1 from dad, 23 times. So we have 23 pairs of chromosomes. Those 23 pairs of chromosomes swapped over or crossed over and exchanged genetic information, and now we have 4 different gametes by the end of meiosis. Once again, this is crossing over, also known as genetic recombination.